Welcome to Cryo Talk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific, featuring conversations between your host, Ava Amson, and experts in the field of cryo-electron microscopy.
Today on Cryo Talk, we're joined by Ariane Briegel, Professor of Ultrastructural Biology at Leiden University and co-director of the Netherlands Center for Electron Nanoscopy. She talks about her research on bacteria in the Hawaiian bobtail squid.
This animal has a light organ inside, and in this light organ, they take up just one species of bacteria that helps them glow in the dark.
Challenges along her career path.
It was really tough. It was this year with the recession, so not many universities wanted to invest a lot of money, and cryo-EM is not the cheapest of methods, so it was really hard.
And how she spends her spare time.
I started going horseback riding again, so I did a lot of that as a kid, and during COVID I decided I'm going to go back to that.
All in this episode of Cryo Talk. Hi, and welcome to Cryo Talk. I'm Ava Amson, and I'm here today with Ariane Briegel. Ariane is Professor of Ultrastructural Biology at Leiden University and co-director of the Netherlands Center for Electron Nanoscopy. Her research group uses cryo-electron tomography to study how microbes respond to their environment. So Ariane, how are you today?
I'm great. Glad to be here with you.
Yeah, thanks for joining us. To start, could you maybe tell us a little bit about your career so far? How do you end up in Leiden?
Yeah, so my career started studying biology at the LMU in Munich. And there, I actually had my main topic was zoology. Actually, I always wanted to be a marine biologist. And starting studying in Munich, it's not the ideal place if you want to do marine biology because you can't be further away from the ocean than actually in Munich. But the zoologists had a passion for marine biology, so they organized excursions to Egypt and other countries to go diving. So it's like that was a big draw. So I started my master's there. And the focus of this group was studying the ultrastructure of crustacean and insect eyes. So they did electron microscopy. So I started working with this microscope, and I became fascinated with this technique. It was just amazing that every time I put something in a microscope, like I saw something that no one else has seen before. So once I finished my master's, I decided this is what I want to do with my career. And not too far from the LMU, we had the Max Planck Institute for Biochemistry, where Wolfgang Baumeister, a pioneer of cryo-electron tomography, has his group. So I joined there as a PhD student. And that's where, what I've been doing ever since.
So is that where you started looking at bacteria and bacteria behavior, I guess?
Exactly. So when I joined the Baumeister group, I think I was one of the first biologists, PhD students that started using tomography to see what we can actually do with this technique and microbes, which is interesting because I'm not by training a microbiologist, but during I think the last 20 years, I hope I became one. So so I started working on that topic there, and then when I finished my PhD, I moved to Grant Jensen's lab at Caltech, who really focused on microbes and their ultrastructure.
Ooh. So why cryo-ET? What makes it so good for looking at bacteria?
Like if you want to look at bacteria, and especially in their entirety, you really need the three-dimensional structure of them. And you can't do it by any other technique, I think, in that sense. So tomography allows you to really look at bacteria in 3-dimension and macromolecular resolution. So you really can zoom in and see each individual cell and see what they have inside.
And then you're looking at interactions between microbes and their environments. How do you do that?
Yeah, so my one of my key research focus in the past 20 years was studying how microbes sense their environment, like these chemoreceptor arrays that they have. So these are basically the bacterial nose. These are large sensor arrays with thousands of receptors that cluster together and form this amazing nano machine that the cells use to sense attractants or toxins in the environment. Since starting my group here in Leiden, I've expanded somewhat more beyond just the chemotaxis, but also how bacteria interact with their environment on a structural level.
And you're also a co-director of the Netherlands Center for Electron Nanoscopy. Can you tell us a bit about that?
Yeah, so that's actually the reason why I'm here in Leiden. So we have this cryo-EM center here. So it's the only place in the Netherlands that has Titan Cryos instruments. So these high resolution microscopes that we need for tomography, but also for single particle. And so we have the center here. So actually all our Dutch cryo-EM community, when they want to collect on the Titans, they come to our center.
Okay, so also from other institutes in the country.
Yes. Yes.
And they, I guess they have to sign up for time on the instruments, or?
Yeah, so the way it works here in the Netherlands, so we many of the universities have their own entry-level or mid-level microscopes, like 100 or 200 kV instruments, where they can do their screening and also some data collection. But then for the 300 kV instruments, they all come with their samples here. And we're supported by a Dutch Research Council grant that subsidizes Dutch users to come to our center. So, and we try to make it very easy for Dutch users really to come here and access the center and collect their data.
That's great. And I guess it's a small country, so it makes sense to have like one central facility for everyone. So what kind of research do you see? What kind of research are people bringing to the facility? What do they use it for?
It's very diverse. I mean, we have, of course, like I think the biggest user group is still people coming for single particle work, so studying protein structures at high resolution. But I think our center is quite unique, that also our tomography community is quite large. So I think I'm not quite sure, about 40-50% is tomography. Another advantage maybe of our center is also we have a biosafety level 2 microscope, so we can also image pathogens, which is not the case everywhere. So
yeah, that would be useful. So what do you think would be next for cryo-EM in general? Like where do you see the field going? Like what advances or applications do you think we can see more of in the next few years?
Yeah, I mean, I think, well, this is not my specialty, but I think for single particle, I think this extreme high throughput that we see in the recent years, I mean, that will allow, like much larger projects like drug screenings, for example. So I think that will be quite exciting. And for tomography, I think, going inside cells is, already a big thing, but like I see the next step. Like I really hope that this large volume acquisition, I think that is where I'm most excited about. Not only seeing like, maybe some cells grown on a grid, but like really figuring out how we can make this technique available to study, for example, pathogen interactions within a tissue, like really go into the real biology of where things happen, right?
Yeah. So are you already thinking up like possible future research ideas?
Yeah, we are actually supported by a Moore grant to do just that. So so one of the things we're studying is the interaction of a symbiosis system. So we started working with Hawaiian bobtail squid. So that might sound like a very exotic animal to work with, but so the interesting thing about this, so this animal has a light organ inside, and in this light organ, they take up just one species of bacteria. that helps them glow in the dark, basically, and hides it from predators from below. So it's only a tiny little squid. It's only like this big. But it's a single bacteria, single host interaction. So it makes this microbiome interaction so much simpler. It's just one bacterium, one host.
Okay.
So we have this grant funded to try and see if we can study these interactions with cryo-EM. So we're trying to figure this out, how to make this possible.
No, that would be so interesting, like this symbiotic reaction, collaboration between a bacteria and a squid, a little squid. And what do you do when you're not working in your lab or in the facilities?
So I like gardening. I like hiking. Mm-hmm. And I started going horseback riding again. So I did a lot of that as a kid, and during COVID, I decided I'm going to go back to that. So
it's so
I
fun.
to go horseback riding.
Yeah. So is it? Are there any stables close to you? Is it easy to get to horses?
Yeah, there's lots of stables. I actually go a little ways out of Leiden. Because my friend who also started horseback riding, we go to her town to go riding.
Yeah, it's one of those outdoor activities that everyone during COVID was starting to get more outdoors. It's been good to get outdoor things back on.
Exactly.
Do you enjoy traveling? I know that's another kind of thing that we couldn't really do the last few years, but
yeah, I do enjoy traveling a lot. So I do like to visit other places and, try other food and talk to a lot of different people. So I do enjoy it.
Do you have any favorite places?
No, I like a lot of places. Recently, before COVID, I visited Japan on a tour. I gave a seminar tour at different institutes, and that was fascinating. I really enjoyed that.
Yeah.
I also like to go back to California and get some sunshine there. Eat some Mexican food.
How long were you there? You did a postdoc there? Or?
Yes, so I stayed nearly 11 years.
Wow, yeah. Yeah, that's a nice place to visit too. And do you like reading?
I do like reading. I haven't done that much recently. When I come home from work, I'm usually too tired to read anything else. But on my vacation, I love reading.
Is there anything you read recently that you'd recommend?
What did I read recently? Yes, actually, I read a really nice book, Remarkably Bright Creatures.
Sounds interesting.
It's a fascinating book, and it's about an octopus. I recommend it. It's really, really interesting and
so
nice.
It all comes back to marine biology somehow. And were there any films or TV shows that you enjoyed this past few years?
Yes, I enjoyed Wednesday a lot. So that was very entertaining. Also, I like a lot of The Lord of the Rings, for example, I enjoyed a lot.
It's funny that you mentioned Wednesday, because you're not the first one to bring it up on this podcast. So.
It cracked me up. I really enjoyed it. Can't wait for the next season.
And you mentioned that you like gardening. Do you also like cooking?
I do like cooking if I have time, but I have to admit I don't do much cooking because I have a husband who is a passionate cook. So I usually sit there, drink my wine, and let him cook.
Yeah.
That's ideal.
That sounds like a good deal. If he enjoys it more, why not?
Exactly.
And what about music? Do you have any favorite music to listen to?
Oh, I'm quite open. So I'm not very specific about the music I hear. I often have the radio on, and maybe the classics like Bruce Springsteen or something like that.
Yeah, can't go wrong with the classics. And I mean, we talked about you wanting to originally be a marine biologist, but what if you were not a scientist at all? What would be your alternate path in life?
That's a good question. So I think before I decided to study biology, I was tempted to also maybe study art. I used to really like all kinds of art, drawing, clay modeling, everything. But I decided science is still my plan A.
And it's still kind of creative. You get to come up with new ideas. Yeah.
I'm working with images, so that hasn't changed.
Yeah. And do you have any advice for some of the early career researchers that are listening? So what would you recommend they do in their careers?
Yeah, so I think maybe the one thing that has helped me the most is like just go with my gut. Mm-hmm. Because when I was finishing, for example, my master's, I talked to a TA in a course I had to take, and he was asking, so what do you want to do after you finish? And I said, I want to go into electron microscopy. And then he's like, so what do you want to do except driving a cab? Like, no one's doing that. And it's like, no, but this is really what I want to do. So luckily he wasn't right. So I think sometimes you just have to follow your gut and not what everybody tells you, like this is important, this is hot, like just whatever you enjoy, just trust your gut.
And in your own career, has there been any surprising turns or events along the way?
Oh yeah, I mean, you never know. Like now I have a lab growing squid. I would have never thought I would do something like that. Yeah, also like there's many surprises all along the way, and it's, I think, never really straightforward. At some point when I was doing my postdoc at Caltech, I started going on the job market, and it was really tough. It was this a year with the recession, so not many universities wanted to invest a lot of money, and cryo-EM is not the cheapest of methods, so it was really hard. So I decided, okay, I'm gonna change my focus. I'm not gonna become a PI. I just want to become a staff scientist, and that would have been done for 5 years until I decided, maybe I do want to become a PI after all. So it was not a straight line.
Yeah, that's always good to hear these stories, that they aren't exactly straightforward and you can't really plan ahead. Is there anything else you wanted to share about your research or about cryo-ET experiences that you think people might want to hear about?
I'm not sure, except maybe that it's really, I think, still starting. I think the excitement it's just building. So I'm just very excited to see where it's going. It's it's developing so fast, and if you see what all the different groups are doing, it's amazing. It's going to be a wild ride in the next few years, I think.
Yeah.
So in terms of like image analysis and also artificial intelligence is a like a key word that many people like to use, but like this automative data analysis and segmentation and all of this is just, I think, coming together, and like will open up a whole new era of biology. So,
yeah, definitely. I keep seeing it everywhere now.
It's amazing. I think it will also like open up to other disciplines, like for example cell biologists who are not they're not structural biologists, but once these techniques are really more routine, they will be very useful for other disciplines as well. So it will be exciting, I think.
Yeah. Well, lots of exciting things to come. So thank you very much, Ariane. I think that brings us to the end of today's episode. And thanks everyone for listening or watching CryoTalk.
Thank you.
Thank you for listening to CryoTalk, a bite-sized bio podcast sponsored by Thermo Fisher Scientific. To view all audio and video recordings from this series, please visit bitesizebio.com/cryotalk.